Afternoon and welcome everyone. My name is John Draper and I am the Vice President of Research and Training here at the Stem Cell Network. On behalf of the Stem Cell Network, I'd like to officially welcome you and thank you for joining us for the webinar today, which will focus on optimizing your publishing strategy. For those who are not familiar with the Stem Cell Network, we are a national not-for-profit organization that supports three main objectives: stem cell research, training the next generation of highly qualified personnel, and supporting the knowledge mobilization and transfer of stem cell and regenerative medicine. Stem Cell Network is committed to empowering early career researchers, that is, investigators who are within their first 5 years of their first academic appointment. We're empowering them by offering structured programs designed specifically for them, covering practical components of managing a lab, such as human resources, budgeting, project management, grant writing, and publication strategies. Indeed, for ECRs, a strong publication record is crucial in success for securing funding and tenure positions. The sessions today will provide you with strategic approaches and practical guidance to accelerate manuscript development and improve publication impact. Our speakers, Christine Mummery and Natasha Chang and Michael Rudnicki, will share their experiences and insights, and a panel discussion will provide you with an opportunity to ask questions relevant to your publication goals. I'm confident that this session will be useful and relevant, not just for ECRs, but also researchers across our network. So with that, I would also give you the opportunity to keep an eye out for another ECR-specific initiative at SCN. We're launching the ECR committee. It launched in January of this year, and they've been busy planning the ECR symposium, which we held in Montreal on May 25th. If you can go to our website, you can then save the date there on our website, and you can also start applying to participate in that. The ECR committee will also be developing other professional development opportunities throughout the year, so keep an eye out for those as well. And so just a couple of housekeeping notes before we get started. Each speaker will talk for 10 minutes, and at the end of the talk, we can take a couple of questions through the Q&A chat box. And then after the talks, we'll have 20 to 30 minutes of panel discussion, and you're welcome to enter your questions for that at any point through the chat box, and also obviously during the panel discussion. This is for you, so please do engage and put your questions in there. This is an opportunity to ask those questions that are really going to help you succeed. So I'm going to begin by saying thank you to all our speakers. I'm excited for what I'm sure will be an informative session, and I'll introduce Michael Rudnicki, who's our first speaker, and who will talk about publication strategy from a senior career researcher perspective. So just briefly, Michael Rudnicki is a senior scientist and the director of the Regenerative Medicine Program at the Sprott Center for Stem Cell Research at the Ottawa Hospital Research Institute. He is a professor in the Department of Medicine at the University of Ottawa. And in addition to his academic appointments, Dr. Rudnicki is also the scientific director of the Stem Cell Network, providing direction and leadership for our research programs. Dr. Rudnicki's achievements are many, and he's a Tier 1 Canada Research Chair, a Fellow of the Royal Society, and an Officer of the Order of Canada. He's internationally recognized, a leader in molecular genetics and regenerative medicine, particularly in the field of muscle development and regeneration. Dr. Rudnicki is a member of the editorial boards of Cell Stem Cell, Journal of Cell Biology and Stem Cells, and he is a co-editor-in-chief of the journal Skeletal Muscle. Dr. Rudnicki has organized many international research conferences, and he's also helped with our TMM conference, which runs every year, you're all familiar with, and was founding director of the Society for Muscle Biology. Michael, I'm out of breath with all of your achievements. I will hand over to you. Thank you.
Okay, I shall share my screen now, get the slides up. Here we go. And there we go. All right, so welcome everybody, and I hope this will be a helpful discussion for you. I think many of the thoughts that the panelists will be expressing are probably self-evident truths that many of you should know already, but I think it's important to enunciate them and discuss them. And I expect we'll have some differences in some of our ideas as well. I've written publish or perish tongue in cheek below that, but ultimately, I'm afraid if you don't publish your work, if you don't communicate what you've done as a scientist, you may as well have not done it. It's essential, not only for your career, but as a scientist, that your efforts to communicate and document and put on the record your scholarly contributions is, that's part of what we do as scientists. And if you're not doing that, you're not a scientist. And that's why so much effort is by our people concerned with promotion and grants and so on, is paid to our productivity. That's core to being a scientist. And publishing your first paper is incredibly challenging. I mean, when you're setting up your lab, recruiting your first students, maybe bringing a technician, maybe a postdoc in the lab, getting your first grants in the door, getting your first publications out the door, and your first research publication in particular, as Natasha will tell us, that's a lot of stuff going on all at the same time. Your kids are little, it's crazy busy time, crazy busy. It's also invigorating and a lot of fun, but you need to be good at multitasking. So it can be really challenging. So I'm going to give you a series of thoughts about getting your papers out the door, sort of a Joycean rambling voyage through where I can provide 2 bits here and there. And just some thoughts about getting your first paper out the door and why it's so important. I think, when I got my first CHR grant, it was for 3 years. So it was incredibly stressful. Everyone, the granting councils in those days gave 3-year grants and hardly ever gave out 5-year grants. Now it's 5 years, but so it's a little better, but I think the expectations by journals of what it takes, what constitutes a paper, has also inflated. So you need a lot of stuff in papers in order to get them into reasonable journals. And I think this is true, but in my judgment, for people coming in with their first grant renewals, so they have 5 years of funding, about half of them have yet to get anything out the door yet. And that really is quite harmful when their grants are being evaluated. But a quarter have had something submitted or posted to bioarchives in revision, and about a quarter have papers that have been accepted and are published. If you can get a papers out the door and accepted and published, you have a tremendous advantage in renewing your first grant. And certainly at the NIH, not getting a paper out the door can be fatal, because you're now no longer a new investigator with a new grant going into a special rating. You are competing with everybody else. Established people are putting out 10 papers plus a year. They're coming in to renew their grants, even though they have multiple grants, and it's not a fair comparison. You need to get your paper out the door and hopefully accepted before you go in for your first grant renewal to be successful, to ensure you're successful. And a research paper really has to be your first priority, and you need to be pushing at that all along. However, don't forget that from the beginning, you can be putting out mini reviews, book chapters, commentaries, methodology, anything like that to add to your productivity. That padding your CV with those sorts of papers also doesn't hurt, but without the research publication, of course, it won't be as impactful. But if you have that and a research publication, I think it just makes you more competitive. And of course, those other sorts of papers, those non-research papers, you can be working on all along from the beginning, where you can. As a young investigator, you may not be, invited to do this at a top journal. That's okay. Just get them out the door and use your network to identify ways of moving forward, to find places to publish this kind of thing. I think working efficiently is something you really will have to learn as a new PI, especially. a lot of people at the bench kind of like starting things, doing the new interesting experiment, but maybe aren't so good at finishing. And finishing is critical to be successful in this business. And also, I mean, you know, we want to be as perfect as possible, but perfection, you approach asymptotically, you never are perfect. And every paper is a snapshot of a moment in time, and things change as you move forward. You gotta finish, you gotta get it out the door. So by working efficiently, also, I think making a plan. What is this paper going to look like? I mean, you have to wait enough time that you have a good idea of what this paper is going to be about and what the take-home message is going to be. That might change with the last few experiments, of course, but make a plan. Think about what the papers will, the figures will look like. Draw up the figures as the data comes in. That helps you identify gaps to finish off critical experiments to go into that paper. And then stick to that plan and get that paper finished. Get your colleagues to read the paper, pass it around. I mean, an early draft, and get their feedback. That's also very important. That also helps identify gaps or problems with interpretation. It's okay to be criticized. It's very, very helpful. That's also true for grants. So get lots of feedback. I think having an authorship policy in the lab is also important. And I think we can discuss that at the panel discussion in more detail. Christine Mummery actually raised this point, and I think she's going to speak more about this. But people have to understand that authorship isn't finalized until the final paper goes in and is accepted. It can change, especially during a revision process. Of a year and a half of work has to occur. And I have had examples in my lab was that there was more work involved in doing the revisions than there was in putting together the first manuscript. And if that person has left the lab, I mean, things can change. Also, if you're going to be an author, you have to have contributed something that's going into the paper. Gratuitous authorships should be, just don't allow those in the lab. I had, when I was a new PI, a student and a postdoc struck a deal without my knowledge that they would put each other on all of the on their different papers, you know, handshake, wink, wink. And they were furious with me when I told them, what are you talking about? You actually have to do done an experiment. That's the work to be an author on a paper. And there's also such things as authorship policies of journals that also inform this. But having a written policy that everybody understands can be helpful in dealing with issues as they arise, and we'll be hearing more about that. I think as a PI, one of the hardest things to learn, especially when you're new and nobody knows how to do anything and nobody can write, is delegation. But as you bring students along and postdocs along, you should be getting them to review papers, writing their fellowship applications, studentship applications, and providing feedback on their writing, and helping them learn how to write, learn how to draw out figures and so on, and get people to do heavy lifting to contribute. If they're going to be first author, they need to do a lot of the work. In fact, they should be writing that first draft of the manuscript. However, if they can't write, and you need to get that out of the door, you need to roll up your sleeves and do it yourself, because you're the one who's going to pay the price. But if you can learn to delegate and train them so that you can delegate, that just accelerates the whole process and moves things along. I think learning delegation as a leader is something that's critically important as you go along. Picking a suitable journal. Now, everybody wants to be in a top tier journal. And that continues through our career. Of course, what is it? The perceptions of top journals have changed over time, and we're not supposed to consider impact factor or other sorts of things anymore in evaluating productivity. But getting into one of those journals certainly is very helpful, especially if you're doing grants and so on. But for your first paper, the problem is that you can get trapped, especially if it's a nature journal, in a 2-year repetitive series of reviews that may not end well, and endless reviews, endless experiments trying to deal with reviewer number 3, and so maybe aiming a little bit lower to expedite publication isn't necessarily a bad thing. However, the paper may be worth giving it a shot, but speed is of the essence, especially if you're about to be renewing your first grants. You certainly should be posting bioarchives when you submit your first paper. This is important for visibility. Some editors will check that to see how many times it's been downloaded, that kind of thing, not always. But you can even publicize it in social media and that kind of thing, and others might cite it. And it's also important for priority reasons. And I think more and more people are doing that, and I think you should give that careful consideration. You need to understand which journals are predatory journals and which journals are crap journals, and avoid them like the plague. Your reputation is everything. And if you start behaving like a bottom feeder, you'll be considered, that will hurt you and your reputation as you go forward. We all know what predatory journals are. There's lists of them on the web. You can Google that if you're not sure. Certainly at our place, we have a publication officer who studies this area, does research with this area, and they help people understand what is and what isn't a predatory journal. Avoid them like the plague. Okay, through the review process. Well, if you're rejected outright, and that happens, happens more often than we like to admit, reflect if the reviewer has identified a critical gap. If all three say this experiment didn't make sense or doesn't support, you're missing something critical, reflect on that and whether or not you have the bandwidth and time to go back and do something. May not be the experiment they suggest that addresses that gap, and then get it out the door as quick as possible to another journal. You may want to drop a notch again to expedite things because of time, or may not, or maybe that new data brings it up a notch, but you'll have to go elsewhere. If you're invited to revise, I would suggest, rather than picking a fight in your rebuttal letter and being with an angry or snarky tone, sarcasm, avoid that. Same with grant rebuttals. It's very tempting because some of the reviewers' comments can be utterly nonsensical, off target, or misinformed. They didn't read the manuscript, didn't look at the data. It's sort of a normative treatment of your paper. Reply to each and every point in a positive way. Many of those things can be addressed in a positive way by clarifying the text, modifying the text in either the discussion or the results section, or explaining better the experiment, but try to do it in a positive way. You can get multiple people involved in the lab to address those experiments, especially, for these first few papers, all hands on deck, and get those experiments done. And if you need to get back to the lab, roll up your sleeves and do it yourself, go for it. I mean, again, it's your neck on the line. And some things you won't be able to do because it's just unreasonable to generate 5 new targeted alleles of your gene of interest, or this kind of thing. And so, with all due respect, you can reply, you can suggest that it wasn't possible to get this done given the time constraints, et cetera, et cetera. And that happens. That's okay. So, if you can get half of the stuff addressed experimentally with new data, that would be terrific. It may not be 100%. If you can get 100%, even better, but it's probably not possible. Often the editors will provide a written summary of what are the critical things you need to do. That's very helpful. If they haven't done that and it's not clear, it's okay to reach out to them and ask them for some guidance. They should reply. They won't always, but you can ask for clarification if it's not clear. I think networking is really important for expediting publication. You need to attend research conferences in your area. This raises your profile. And I don't mean, the ASH where there's 10,000 participants. I'm talking about Gordon conferences, or those smaller conferences where you actually spend time on site, eating together, having a few drinks together, and getting to know people. Build your network. And many of those people who are at that conference will be reviewers of the paper. So grab them and bring them to show your poster or show them your data on your laptop. You know, getting out there and socializing and working that network and forming connections, starting collaborations if possible, all that is really helpful to raise your profile. And when these people get your paper to review, they've met you, they can put a face on the name, and that just helps, because this is a human endeavor after all. Often editors of big journals will be attending meetings. So again, network, introduce yourself, tell them what you're working on, see if they're interested in what you're working on. This can be very helpful to have relationships with these people, and they also can move from journal to journal, and that network can expand as your careers goes on. And even better if you can have a few drinks with them and a laugh and get to develop some rapport. That's not always possible, of course. And that wraps it up. Thanks very much.
Fantastic. Thank you, Michael. If anybody has any questions at this point, do drop them into the Q&A. I'm keeping that open. So if not, then I will move forward to introduce Natasha. So Dr. Natasha Chang received her PhD in biochemistry from McGill University and pursued her postdoctoral fellowship at the Ottawa Hospital Research Institute. Dr. Chang joined McGill as an assistant professor in the Department of Biochemistry in 2019. Research in the Chang laboratory focuses on understanding the molecular signaling mechanisms that regulate muscle stem cell function and how these pathways are altered in the context of muscle degenerative diseases and muscle cancer. The ultimate goal of Dr. Chang's research is to identify molecular targets to improve endogenous stem cell regenerative capacity, as well as strategies to improve stem cell transplant therapy. So Natasha, please go forward with your ECR perspective on publishing.
Thank you so much, John. Can you see the slides?
Yes, I can, yeah.
Okay, so thank you very much to the Stem Cell Network for this opportunity to participate in this webinar. I've been a PI for 3.5 years since 2019. And so I'm still very much in the learning phase as to how to run a lab. And so one of the most, I think, terrifying tasks for new PIs is to take that step and to publish independently, because before, as a postdoctoral fellow, you had always the support of your mentor. And now you need to learn how to navigate this on your own. And so full disclosure, I did my postdoctoral training with Michael Rudnicki. And so there might be some overlap in terms of how we go about and approach things. But for my advice for those who are early career researchers is to really think back to those main first author publications from your postdoc. Because I think in essence, you have already been in training for this, and you might not have even realized it. And so I can remember as a postdoc with Michael, that Michael really would give us the opportunity to put that paper together from start to finish. So when the time came and the story was coming together, the first thing he would ask us to do was to put the figures together. So it all starts with the figures, and I would go through my data and decide how to make figure 1, figure 2, and so on. And then Michael would look at it, and I remember him printing out the figures and then cutting out some of the panels and then taping them back together. And so this was his way of seeing how the story comes together and what's the most linear way to tell the story. And for me, this was actually a really valuable lesson for how to put together those research papers. And of course, we had conversations about where to submit. And it really depends on the nature of the story. So of course, we wanted to aim high. And we went through a couple of desk rejections at top tier journals, Cell and Nature Medicine. And then we finally settled into what I think we had always known from the beginning would be a good fit for the story. And so that's where we ultimately ended up publishing at Cell Stem Cell. And then as first author, I was really responsible for drafting the manuscript. And so we all know this process that I included a picture of it on this slide, which is that endless cycle of going back and forth with your mentor. But all of this is actually really valuable training. It's training you how to write and how to be critical of your own writing. And then finally, Michael would involve us in all the steps during the submission process. So I remember drafting the cover letter, which he, of course, made a lot of corrections to. But to this day, I still look back on that cover letter and I use that as a model for writing my own cover letters. And of course, when the reviewers' comments come back, we would sit together and devise a strategy for the revisions. And again, he would give me the opportunity to write the response to the reviewers. And every PI has a unique style for approaching this, but I think this has really impacted the way that I write responses to reviews, and not just for manuscript, but also for grant resubmissions. And so what I think I'm trying to say ultimately here is that most likely you are more ready for this than you think, and that you've actually had a lot of training for writing and submitting manuscripts. And so you already have the tools to do this as an independent PI. And certainly these experiences as a postdoc has shaped the way that I've handled this process as a PI in my own lab. And so as a new PI, we are faced with a lot of challenges, and we're overwhelmed with a whole new set of responsibilities that we didn't used to have. And of course, there's this pressure to perform. And so for those of us in tenure track positions, we know that the research component is the most important part of the tenure dossier. And of course, there is this pressure to publish. And so my advice here is to really balance your commitments and find ways that work for you to manage your time. So not only are we setting up the lab, writing grants, trying to recruit students and staff, but we are also asked to review manuscripts, and we're also invited to write and contribute papers. And so while I think it's important to have those publications coming out of your lab, because this shows productivity, it shows that your lab is running, and grant review panels really like to see this. It also takes time away from your research. And so there needs to be this balance so that you don't stray too far from getting your research program going. And so the very first paper that I submitted as a PI is a review article. And I was invited by colleagues who were putting together a collection on stem cells and metabolism. And because I knew the scientists who were inviting me, I decided that this would be a good opportunity to get my name out there and to really carve out my research niche. So I wrote the review on autophagy and stem cells, and it was a lot of work because I didn't have any students at the time. This was right when I started, but for me, this was a way to tell everyone, this is what my lab is interested. And when people apply to my lab, they have seen the paper, so they know what they're getting into. And once you have students in the lab, you can then delegate and get them involved. And I find that students are always really eager and excited to contribute. But I have found that at some point you need to start being selective, because you will receive invitations and you can't possibly say yes to everything. So how do I decide what to take on? It depends on many factors, but it comes down to if I'm passionate about the contribution. Will it be beneficial for the field, and really who the invitation is coming from? As Michael mentioned, we are all aware of predatory journals. This is actually a problem that I didn't realize was so big until becoming a PI, but ultimately we want to publish our work in reputable peer-reviewed journals. So do background check on the journal that invited you. And again, balance your commitments, make sure you have enough bandwidth and time to take on a new project, and make sure you have students that are willing to take on the task. And so actually, two of the papers that have come from my lab are actually methods articles that were part of a stem cell network initiative in collaboration with Current Protocols. And that was to expand their collection on stem cell related protocols. And these were actually really fun for my lab to do because it allowed us to collaborate with other labs to gain expertise. So we did this for one paper on immunofluorescence imaging and quantification. And it really brought the lab together in a collaborative way to optimize and troubleshoot these protocols that we are using routinely. And so this brings me to my next slide, which is to use these situations as an opportunity to involve and educate your trainees. So just like how we learned as a postdoc how to put papers together, now I think is the time to pay it forward and train the next generation. And so at a time when you're just starting your lab is pretty small. And so you have a handful of students, you can really provide everyone the opportunity to contribute. And I've always tried to include the people that were in the lab at the time for every publication. And this includes both undergraduate as well as graduate students. And as the lab got bigger, for example, one of those methods papers, I had a very open discussion with the lab to explain what are the responsibilities of the first author. And I asked who wanted this task. And so I think communication and transparency are an integral part of how I move forward with publishing and how I basically try to run the lab in general. And I feel that with each publication, I am always learning something valuable about how to manage a research team. So it's really a learning process for myself as well. So some of the tips that I've learned is to set deadlines and be very clear about delegation. So who is doing what. Regular meetings as a group is also really helpful to make sure that everyone is on the same page and that everyone can see the direction that we are taking. And so we're actually using this strategy now as we plan out our research articles that we're hoping to really submit this year. So we discuss them at lab meetings, and we take this on as a group to see what are the experiments that are missing and what we need to do to complete the story. So ultimately, I think this activity really helps to build the research team and to establish the culture that you want to have in your lab. And so finally, I just wanted to make the final point about promoting your work. I feel really strongly about publishing open access because if people can't access it, then your work doesn't get read or acknowledged. But there are other ways of getting your research out there, such as using social media and platforms such as Twitter. And so you'll often see people posting these, they're called tweetorials about their work. So this is an example of a recent paper that came out of my colleague Wahab Soleimani's lab. And he talks about the significance of their findings and how they came about them. And so they're basically the cliff notes of the paper, essentially. And of course, making sure your Google Scholar profile or ResearchGate profile with all your publications is up to date is always good practice because many of these people use these search engines to find your publications. So I'm gonna end here. I hope I've demystified the process a little bit for new PIs. I think when I was preparing for this, what I realized is that we're actually more trained for this than we might think, and we can utilize this process as a training opportunity for our own labs, and at the same time be, of course, contributing to scientific knowledge.
Thank you very much, Natasha. If there are any questions, do put them in the chat. And I would actually point out, Natasha alluded to the current protocols opportunity. That's still open. And if you look in the most recent Stem Cell Network Cell Lines email, you'll see that there's a link to that. So if you're interested in using that as an opportunity to publish some of your protocols, then please do connect either with me or just look at the opportunity on the website. Our next speaker is Dr. Christine Mummery. Christine is a professor of developmental biology and heads the IPSC hotel facility at the Leiden University Medical Center. She trained in biophysics at the University of London and since 2015 has been a guest professor at the University of Twente. Christine's research focuses on modeling cardiovascular diseases using stem cells from patients. on developing organ-on-chip models of multiple organs for safety pharmacology and potential disease and drug targets. She leads a multi-million NWO Gravity grant for this purpose and holds European Research Council Advanced and Proof of Concept grants. She co-founded the European Organ on a Chip Society and the Netherlands Human Disease Modeling Technology Organization. Most recently, she became part of the Novo Nordisk Foundation project, Renew, on human stem cells, which links nodes to the Leiden University Medical Center, Copenhagen and Melbourne to advance regenerative medicine and drug discovery. She's a member of the Royal Netherlands Academy of Science, a past president of the International Society for Stem Cell Research, and was the founding editor of the official ISSCR journal Stem Cell Reports. She has sits on several scientific advisory boards, including Hubrecht Institute, Gurdon Institute, Allen Institute, Mogrify, and Sartorius GmbH. Christine, please take it away with your expertise in being an editor. Or you are on mute, Christine.
Okay now?
Perfect, thank you. And I can see your slides as well.
Okay, perfect. Yeah, so I'm going to talk a little bit more from an editor's perspective on this. I was one of the early editors of Stem Cell Research and then moved 2012 to found Stem Cell Reports, and I've seen hundreds and hundreds of papers come through the editorial desk, you could say. So how do you get your paper published? I'll just start off with the way many people see publishing as an undergraduate. You think you start a paper, you write a paper, you submit it, there's a decision, and you get it published. However, when you get a little bit further as a grad student, it doesn't quite work like that. So you start a paper, write the first draft, find an error in the analysis. Sorry for the noise, somebody at the door. You edit the paper, you send it to your supervisor, and you wait. And then when your supervisor has given you feedback, you send it to your co-authors, and you wait, and you edit. And then you submit, and you wait again. And then you get a decision. Now, these decisions could be reject with the opportunity to resubmit, accept with major revisions, or accept with minor revisions. Now, reject is reject. That's very hard to fight against. It's not only what the reviewers might have said, it might be the impression of the editors, and it might be what they said off the record. Now, we don't really care for that mode, but nevertheless, it can happen that the reviewers will say something to the editor that they doubt the quality of the work. Reject with the opportunity to resubmit, you have to do a lot of work. Accept with major revisions, you get about 3 months to do it. And many journals will let you have one shot at goal, another shot at goal, and so on. And one of the things when you write your first draft, for example, you can get caught up in an exponentially growing literature review. It can be much too vast. You're not writing a review paper, you're writing what's necessary to introduce the work. And you can get writer's block. So many people are better off starting, let's say, with the materials and methods. Then you don't have a blank sheet of paper, you've got something out there. Go to the results, then go back to the introduction, finally go to the discussion, and then back and write an abstract, because then you know what it's really about. Just a suggestion of how to get to it. So what makes a good paper? Groundwork is essential. So the most fundamental agreement, of course, is excellent research. You need to work with the best scientists you can, in the best lab you can find, or have the best collaborators you can find to ensure you're surrounded by excellence during your training. Make sure the questions you investigate are important and of interest to others in the field. If you're working on a totally uninteresting question or is totally unimportant, then it's not going to get very far. The most successful papers are those that present innovative research, but the best papers also present their story in a clear and logical way. It's very straightforward. The thinking behind the paper must be clear, so the writing is clear if you have a story. Writing good research papers can require a bit of strategic thinking, practice, and lots of know-how. But remember, practice makes perfect. So beginning with some small, what we would call perhaps bread and butter papers, a simple description of a new cell line, send it to stem cell research. It's a very short paper that will give you the practice of actually getting the whole process done. The next question, is your story really convincing? So basically the reader must come to the same conclusions as you on the basis of your results. So ask yourself whether the reader will be led to the correct overall conclusion. If they would come to another conclusion on the basis of the results, you have a bit of a problem. You also have to ask in the first write, and that's why Michael's remark about getting the figures together first, what convincing experiment is missing? Better to do it now than later. Also ask yourself, are there alternative explanations for your work, for your outcomes? And if you don't give them and put them up front, the reviewers certainly will. They will interpret things differently sometimes than you intend. So what data would you need to collect to eliminate those alternatives? And before you perform the new experiment, ask yourself how it will contribute to the logic of the publication. You need to test your reasoning on colleagues by asking them whether you told a logical and convincing story after giving a talk, for example. Because if you can't tell it in a talk in a fairly coherent way, then you'll never be able to write it up. It's just about telling stories. So think in figures. Michael said it already. Every experiment you or your students are doing should be fitting in some master plan for figures. Judge when you have enough data to write a paper. If you write too soon, you may be wasting your time. Wait too long, and you risk getting scooped. So that's the problem. Stop and write when the data are sufficient to tell a story that is complete and makes sense. The key is to constantly keep the paper in mind when you're performing your experiments. Think about the figures that can already go into the figure, into the paper, and the information they will contain. Visualizing this is really essential. So, frequent mistakes that we come across when we're editors. So the abstract results, methods of conclusion are not the same as the reported results. That's what happens if you write the abstract too early. Sometimes it doesn't match with what you actually report in the paper. You exceed the word limit allowed by the journal. Just don't do it. Make it within the word limit. It can be formatted incorrectly for the journal. So unstructured versus structured. Sometimes you see abstracts which have an introduction, results, conclusions. Other journals don't have that. Nevertheless, the hidden structure could be the same. And if you're going to a more general journal, the general interest of your work is not sufficiently emphasized. The easiest comment you get from a journal is, should be going to a more specialized journal, but you don't want that. So if they say that, it means you didn't make it general enough for the journal you've chosen. So as an editor, we say, look for the seed of brilliance. So when we read a paper, we're looking for something novel, something brilliant in that story. So if I would talk about stem cell reports, it first goes to our managing editor, who simply looks at the figures. Are they looking correct? Are the stats correct? Is it likely to be somewhat novel? So she knows the literature very well. And then it gets passed to the editor-in-chief, who decides who's going to be the handling editor. And this is with many journals. There is an editor-in-chief, and it passes on to a handling editor. Make sure your abstract captures the novel aspects of your work. So it shouldn't just be in the cover letter, because many journals don't send the cover letter to the reviewers. So it's very important that you have that captured. The introduction. It doesn't describe the purpose and objective of the study. That's what the introduction is meant to do. It doesn't mention the importance and originality of the study. You're not allowed to say, we are the first to. Priority claims are not allowed, but you need to show why it's original. The introduction may contain issues may contain references unrelated to the study. Take them out. Contains material belonging to in other sections of the manuscript. Don't go too far into the details because, of the results, because they belong in the results. And it's simply not interesting. So that can happen too. So our advice as editors is what we call the KISS principle. Keep it short and simple. Even if the journal is for a scientific audience, using simple language to explain complex ideas is a great benefit. So having short abstracts or introductions with a word limit is very, very helpful because it makes you think concisely. Method sections. We find lots of things wrong with method sections. Some methods reported not used. Some methods are missing. Thus, they don't allow duplication of the study. So you may have written the methods early and changed the paper and forgot to go back to the methods and check that they're okay. The reports of the statistical methods are incorrect or they're poorly described. And the methods described don't really relate to the results very clearly. The reported data can be incomplete, contains the results from another study. We've seen that. We thought, what on earth is this about? And it was clearly a copy-paste from another study. The information repeats too much of what's in tables and figures. So let the tables and figures stand for themselves and keep it very short. Don't put lots and lots of numbers in the results section. And some people are tempted to put discussion or methods into the results. Keep the sections clean. Keep the discussion for the discussion and the methods for the methods. That's very important, otherwise it'll get too cluttered. And the discussion, it can be biased because you have a hypothesis you want to test, or it omits findings from other studies. Now, this is very frustrating for reviewers because they would sometimes, perhaps you've omitted major papers that they have written. So don't get them upset by doing that. Look carefully at the literature and make sure you've put your study in a fair context. Maybe you haven't explained the key results. They have to be there up front. Maybe you haven't described the limitations of the study. Better not to overclaim, explain the limitations of what you have not, by the things you have not done. Maybe you should acknowledge the speculative aspects, keep them very clear. Don't include outdated references or misrepresent them. We find many times that people cite an article, and in fact, if you go back to the original article, there's nothing in it that relates to the citation. And sometimes people use outdated references. So don't use reviews unless necessary. Use primary data and use the most recent ones. So if your most recent reference is from 2012, you've done something wrong. You can overstate the importance of the study, or it's too expansive and lacks a logical flow. Think always in terms of stories. And the conclusion, just restates the content from other sections, not a good idea. Includes statements that are not supported by the study. It does not clearly relate the findings to the purpose of the study, and it contains unnecessary information. Again, this has to be very short. And just as a reminder, if you think English is not your first language, seek help from a native English speaker to proofread your manuscript before you submit it. Now, you could say this is not important, but if the reviewer and the editors have a struggle actually understanding what you've written, it's a bit of a shame for your work. So they have to be really concentrated to figure out what this story is about if you haven't done it. So I'm sure you have a colleague who's a native speaker, or at least a good speaker of English. Don't discourage reviewers or editors. And many journals these days don't have proofreading services. So handling the review process, submitting your paper, you can help the review process go smoothly by providing a cover letter that includes in very clear language a concise version of the logic of the paper. So don't copy paste the abstract. It's far too long, just a few sentences. What did you do and what did you find? If there are any special consideration that the editor or reviewer should take into account, include those in the cover letter. If you could say, actually, I've just seen a paper on bioarchive on the same field, you can say that because they may take into account when they're reviewing. Find out if the journal is interested. So if the journal is interested, you have to assume the reviewers are offering their suggestions as constructive criticism. They're not trying to destroy you. It's meant to be constructive in general. And the editors will often not send or will edit very damning reviews because we don't want to destroy people. Make all possible attempts to comply with their requests, including performing the extra experiments, even if you think they're unnecessary. You might want to send data in just for the reviewers, and don't be afraid to ask for more time. Many journals these days have what we call scoop protection. So if it's in revision, you are protected from scoop. EMBO Journal does that, Stem Cell Reports does that, and multiple other journals. One important clue, though, is choosing the appropriate journal. It's been covered by others. Choose the most appropriate journal and save yourself much effort and time and get your results into the world sooner. It's always tempting to send your paper to a top journal, even if your results are not groundbreaking, but you can save a lot of time and reduce your frustration if you send it to the appropriate journal first, instead of waiting until it's rejected by a top journal. And they can take a very long time to reject. We've had papers, top journals, sometimes for 9 months before you get a clear decision. And people, I know somebody who's actually had a paper at Nature for 5 years, going round and round and round, but a single figure is the same as when he submitted it. The so-called top journals value novelty and unexpected findings. They may not always be correct findings. They want something unexpected. But other journals may be interested in and appreciate careful, extensive analysis of critical processes. So one of the biggest compliments I had ever was from a top scientist who said, "Christine, if you publish it, I believe it." So you could be in that category. I think that's a very wonderful thing to say, and I've always valued that comment. In general, journals with editors who are professional scientists are more likely to see the merit of your study, even if it's imperfectly presented or incomplete, and can help you to improve it. So there is an advantage in that. The remit of editors of top journals is to get high citations. It's completely different from the motivation of a scientist very often. And this is my final slide. If there's an opportunity to suggest reviewers, use it. The reason for that is many journals will choose 1 or 2 even of your preferred reviewers. It's very hard for journals to get reviewers these days, and often a suggestion is most welcome. You may also exclude reviewers, and at least I speak for Stem Cell Reports, we never have ever invited an excluded reviewer because they're often competitors. On the other hand, your suggested reviewers might not be your friends or as friendly as you think. So the biggest criticism can come from your preferred reviewers. And check papers on bioRxiv. Your competitors may have deposited their paper under review there. And good luck and hope for good reviewers.
Perfect. Thank you very much, Christine. And whilst we'll move on to the panel discussion, please do enter questions into the Q&A, and we'll just get started with, maybe we'll touch on this point of the bioRxiv and preprint server submissions. Michael, what's your take on the value of a preprint server submission? Where does it position you? How does it protect you? How does it endanger you?
Yeah, so I mean, we've done it on occasion, but we don't do it religiously, I would say, in for my lab, and that probably just reflects where I'm at in my career and that kind of thing. So it certainly is helpful for people applying for fellowships and other sorts of things to document that there is a manuscript and it's on its way. It can also establish priority. I think as a young investigator, though, now it's pretty much ubiquitous to do that. I don't think it's harmful at all to get your paper out there so your colleagues can see it and so on. And certainly, I have seen important papers, languish in bioRxives for 2 or more years before the paper finally comes out. So it's a really nice way to see what's going on that otherwise wouldn't be available. You know, if a paper is a bit controversial or something, it can get held up. But there's some neat stuff that's there. Presumably there's also lots of crap, so you have to use your detector and use your own judgment. But for yourself, when you know, if it's on bioarchives and it's not out yet in a proper journal, that still is helpful, especially for grant reviewers and so on, to gauge your productivity. So I think it's a good thing, and that it should be done, especially for newer investigators.
Natasha,
I'm
sorry.
Thinks about that.
Natasha, is that your experience as well? Do you feel that the bioarchive or a preprint server is it's helpful, or do you feel that it could lead to risk for an early investigator regarding getting scooped, for instance?
I agree with Michael. I think it's more helpful than harmful. And his point about it being useful for grant submissions, I think is spot on, because, you know, it takes so long these days to get a paper published. It can go through many rounds of revision. So I think having something in bioarchive is a stamp to show that you have a publication that is more than just saying in preparation, right? It's actually something in a form. So I believe it's more helpful.
Christine, do you find that because there's no editorial process for a bioarchive preprint, does that endanger, you know, could you give the wrong message potentially? You know, it's a premature sort of account of the science being done.
So in terms of the grants, it does get a DOI, so you can actually cite it in other papers, so it's findable. That's very beneficial, and of course you can update it all the time. So if you got it wrong, I was a bit old-fashioned in the beginning, so I thought, ooh, I wouldn't want to put my secret results out there. But sometimes it's beneficial to actually have claimed the date. So in a very competitive area, you might want to, have some kind of priority. Actually, it was out on our bioarchive first. So I think there are reasons to do it. You might get very useful feedback. And I think we've had that. People say, have you thought of looking at this? And then you get actually a gem of an idea of an experiment you might do. And it's actually benefited the ultimate paper. And so we've, acknowledged people who did give us a good idea on the basis of bioarchive. So there's lots of benefits. I think there is something of a risk if it's really novel, and there's a big, powerful lab who thinks it's novel, they can do it before you. But that is actually, our business isn't always very clean, and everybody knows that sometimes it has happened that reviewers will protract a process and do experiments very quickly, and then suddenly it's a back-to-back article to the original one. So I've had, it's not just stories, these are realities. So why not put it on bioarchive?
Excellent. So we've got some questions coming in, so I'll read the first one. Is it recommended to reach out to editors prior to submission to gauge the appropriateness or interest of the journal in the study? That's from Golnaz.
Many journals offer that as part of their editorial process. In my opinion, and others I'm sure will disagree, you might as well submit the full manuscript, because that's what's going to be judged. And submitting an abstract is not the same as the full manuscript, and it can take a long time to get feedback. So
just prolongs the process.
I think, though, if you're having, if you meet an editor at a conference and show them your data and get them interested or excited about that, that doesn't hurt.
Yeah, so I would agree. I've sort of jumped ahead, sorry, Natasha. But editors are looking at posters, and they will engage with you in discussion. With some PIs, if they begin to get to know you, they ask you, what are you doing? What's going on in your lab? And you just tell them, and they may or may not be interested. But a lot of journals do have a formal pre-submission inquiry system. So they want to see the figures, they want to see the abstract, they want to see the main points, and they will give you some feedback. And that is, it's nothing to do whether the work is good or not. They assume if you've done that, the work will be robust, but it's more, is it fitting in the trend they want to see their journal grow or something like that. Mm-hmm. So I think that there is a benefit in reaching out to the editors if there are opportunities.
Natasha, do you want to add anything?
Just that I think, you know, here again, networking is very important. So you want to meet these editors when you can. Sometimes it just happens spontaneously at a conference or they're visiting your university. So I think those kinds of interactions are probably more helpful than reaching out via email. So yeah, I agree with what's been said.
Okay. Next question from Shin. What do you think of the value of transparency during the review process, like in Nature Communications or eLife? Is anyone familiar with that process?
You mean where reviewers show their names?
I think that's the
anonymous.
Yeah, so you can, with nature journals, you can tick these boxes. Do you want to be known? Do you mind your name being there, but not associated with a particular review? There's a great danger in that in many ways, because, for example, if I put Professor, Netherlands, heart cells and stem cells, it de-identifies you, absolutely. So, and there are other people that happens to as well. So if it's if it's with the name, then it's fine. But if it's anonymous and then you just fill in demographics for them, then that's not very good.
Yeah, I mean, in theory, outing the reviewers publicly would prevent ad hominem, snarky kind of reviews, that kind of thing. On the other hand, it opens the reviewers to retaliation, who are, gifting their time and energy and effort to do this, and most try to do an honest job with it. I think what the system that eLife has previously developed, I'm not too sure about their newest iteration, but where the reviewers would be outed to each other and have a group discussion and then a consolidated review put together. I really like that. Yeah, I think that's a very innovative system, and it's like the grant review process where people are kept honest by being known to the committee reviewing grants. Yeah. The people doing the reviews are known to each other. Yeah. It's much harder to be a jerk as a reviewer.
Absolutely, and it's but it's also labor intensive. That the way eLife did it was, of course, brilliant, perfect, but I think the review system is not sustainable. Yes. So when there's so much being published and you get so many declines of people reviewing, I'm already doing 3 for another journal, and it's true, it's not just an excuse. There's so much reviewing going on. I wonder if, in the end, everything will be bioarchive and things will be cited on that basis. The papers that are good will get cited. People will do their own review process. If it's not good, it just won't get cited.
I totally agree.
Interesting. So, maybe this is a question you can have a go at, Natasha. Would you recommend publishing some of the findings quickly if they have great potential to be translated into, have translational potential? In this case, they're saying biopharmaceuticals, but what's your strategy there? What do you think is appropriate?
So I think as a new PI, you want to publish your first paper as soon as possible. The advice that I've been given by many senior mentors is not to aim very high for your first research publication, just get it out the door. So yes, if there is potential to be for your work to be translated, I think yes, you do want to get that publication out the door, and not to think as a postdoc and think, oh, I need to get that huge paper. I think you want something respectable, but you want it to be a fast process.
I think also in my experience that translation is slow, arduous, and takes much longer than people anticipate. I mean, you know, when a reporter asks the scientists, how long before this will be in the clinic? Oh, 5 years, no problem. And there we are 20 years later, and it's still, you know, being developed. So I'm not too sure that being something that's translational is necessarily going to be faster.
So we've got quite a few topics to move through, so I'm going to move on and ask, and this is a question that I think is going to be of value to many. Deciding authorship, the order, avoiding and settling disputes. Can we have some comments on that? Maybe starting with you, Christine.
Yeah, so most senior PIs sort of have an intuitive way of doing it, but most, if we talk about it with other PIs, they're doing it in the same way. But we've just been discussing, at least in our department, and discussed it recently among the next gen of ISSCR investigators. Does your lab have a publishing policy or not in terms of authorship? And actually, many people do find it helpful to know when they join a lab, what the authorship policy is. So what can they expect? As a PI, it's beneficial sometimes to have a higher impact paper by fusing two projects. But that means very often you have two first authors. So who's going to be first and who's going to be second first? The senior author always has the deciding role, but it can get very uncomfortable. But if you have all these sort of guidelines, you could say, for publishing and authorship in your lab up front, then the way it's discussed is transparent. So it often cannot be an entirely democratic process. The rules of the journals are you have to have had a proper contribution. All of us suffer sometimes. People who in the clinic who provided one bit of tissue, stuck it in ice, and then gave it to you and expect a senior authorship for that. Now, those are very uncomfortable discussions. Or you collaborate with somebody, they do a fancy technique, and then expect 4 authors on a 3 authorship paper. So it's very complicated. But having some guidelines, and as a starting out PI, when you collaborate with somebody, think about these things. What's the impact on your paper? Because it is important. We've had papers where the first 2 or 3 are clear, shared, 3 first authorship shared, 3 last authorship shared, and everybody else in the middle alphabetic. In physics, it's always alphabetical. They never have these rounds like you do in biology. It's amazing. But biology and medicine are slightly different. But having a policy up front can be very helpful. And this point of the authorship's only definitive when the final revision is accepted is the only way junior PIs or PIs in general ever get papers finished, because if you can't offer anybody anything, they work for 2 years on a revision and you can't offer them first authorship or joint first, then you have a problem. You just never get the work finished. And the advice to people moving on is try to stay until the final revision has been finished, because you cannot expect to be first author on a paper if it's published a year after you've left.
Natasha, how are you tackling this in your lab?
Yeah, unfortunately I don't have anything written down in terms of a strict policy, and I think it would be hard to come up with the many different situations possible, because it's dynamic, right? Like a paper can change from draft 1 to final acceptance. So it's hard to envision all of those situations and put it all down on paper. But how I've approached it is to have conversations with my trainees as we're going through it. And so I mentioned that for one of the papers, I spoke to everyone during lab meeting, and I said, this is the methods paper that we are going to put out. This is the responsibility of first author. And I made it very clear what first author responsibilities are. And I asked who wants to take this on. 2 people actually volunteered, and so we agreed that they would be co-first author. And then when it came time to deciding who would be first as co-author, they actually came to an agreement amongst themselves. And so it was a nice experience. But it is clear that as senior author, this is your responsibility to determine the authorship ultimately.
Mhm.
And I've had to, people that have volunteered and said, I want to be part of this paper. In the end, they didn't actually do anything on that paper. And I've had to sit down with them in my office and explain, this is why you're not on the paper. And I think that's important to do.
Mhm.
And people do take it well. So I think it's all about communication and transparency.
Perfect. Open access versus closed access. What's the benefit? Where should early career researchers be heading?
The question's clear. If it's
Open.
CHR or NIH funded, it must be open access. Full stop.
Anything in Europe must also be open access. And of course, the reason for open access, the argument is we pay the journal, the publishers enough for subscriptions and all those things. We work for them for free as reviewers. We work for them for free as authors. Of course, everybody, the research is paid for often by public money. So of course it should be open access.
Yeah,
Okay.
and when you go to renew your grant and it's not open access, you cannot list it as productivity. But every now and then I'm surprised by a little chapter or something where I didn't look into it close enough, and it's really annoying.
We've got another question which says about how to strategize as a junior PI choosing between having constant productivity, i.e. several small papers, versus investing in a bigger story. How do you make that decision?
Well, I think what's important when you're being looked at for productivity is the trajectory. You want to be building over time over your career. You want to avoid long gaps or diminished a loss of productivity and tanking or that kind of thing. And so you want to be building your reputation and the tempo of publications as you, especially over the first 10 years. And then it'll reach some kind of steady state depending on the size of your lab. In terms of judging, numbers of small versus major publications, I mean, that first phase, if you can get a big splash every now and then, that's great. That's probably sufficient. And what's more important is getting stuff out the door to establish that, yes, indeed, you can publish papers, especially for renewing your first few grants. And renewing the second grant, you need to get stuff out the door. Now, doing everything by minimal publishable unit. If you're publishing non-interesting tiny papers with one figure, but in journals that nobody's ever heard of, and doing all your papers that way, that's not a good way to do business. I mean, your reputation is everything. So you want to publish papers that have a take-home message, that have interesting findings that are adding to human knowledge and contributing, having impact. Although I think with Dora, we're not allowed to use the word impact anymore, that people will be interested to read and contribute to knowledge. And so, getting a paper and sell is terrific, but it's not the only way to have impact. In fact, most work that results in Nobel laureates are published in minor journals because they weren't considered, or they were too controversial at the time. It's really interesting. So the most impactful work often isn't published in Cell, Nature, and Science.
And one of the things is, if you have a biggish lab, people might only have a grant to come for 2 or 3 years, and they have to have for themselves some evidence they were ever in your lab. I mean, if they leave with nothing, how are they going to justify their grants to their grant givers?
Mm-hmm.
So having what I call these smaller bread and butter papers, which can be very useful, current protocols, for example. Nothing so good as a good, solid protocol. And they're very interested in having things that work robustly. Nature protocols is also a good way to go if they're interested in your method. I mean, even Jovi, that's very, very helpful.
Mm-hmm.
And everybody knows the journals. So that's for bread and butter articles, whether you're a PI or you're somebody in a lab and think you want to write it up. I think that they're very useful articles. They always get cited and they put you on the map because people know you exist. Whereas if you have to wait 4 or 5 years for your cell paper, even then, they may say, oh, it's a one shot at goal. They got lucky or they worked hard, but just one paper doesn't make a career.
That's a good point. Okay, final question, because I think we have to wrap up soon. Brief reports versus full articles. What are the benefits? Is one easier than the other? Where should you be focusing your time?
I see a lot of benefit in a brief article, so something with 4 figures and a short text. So many journals have a reports format, which is short, to the point, a single message, nothing too complicated. And those single message papers are often extremely useful and are very well cited. So if you've developed something, you found something out, if you're not going to have the bandwidth or time to find out all the different applications of it, might as well put it in a short report, which can be, I think, very useful. If you've got the people and resources to use the method or use the technology to make a big paper, fine as well. Always go for more if you can.
Yeah, I would say it really depends on your figures. So put your figures together and see what's the best way to tell your story. If it's a short report, I don't see any problem with that. But it all depends on the data and how you want to present it.
Any final words, Michael?
I think ultimately you'd have a blend of both kinds of contributions through your career as you move forward.
Okay. And with that, I'm going to say thank you very much to Natasha, Christine, and Michael for participating in this webinar. I hope it's been of use to the ECR community, and we're going to say goodbye for the afternoon, and thank you very much.